Good morning all. Today I am taking a lecture on viruses. In the today's lecture I will include the general characteristics of viruses, classification, lab diagnosis, some information about the bacteriophages and HIV, that is human immunodeficiency virus. First just I want to discuss what is virus. Virus is a term that is derived from the Latin words that means the poison or the venom. In 1886, the Adolf Meyers he has saw this virus in the TMV, that means tobacco mosaic viruses. So we can say that virus is an ultra microscopic infectious agent that are metabolically inert, which multiplies only within the living cells. Okay. Now we can also say is that virus are unique organism, that is infectious agent, whose distinguished is that resides in their simple acellular organization and patterns of reproduction. The complete virus particle, that is called the virion, that is consists of one or more molecule of the DNA or RNA enclosed with a coat of protein, and sometimes that also be in the layer. These additional layers may be very complex and contain the carbohydrates, lipids and additional proteins. Virion can exist in the two phases, that is extracellular or intracellular. Virion, the extracellular phase, possesses a few, if any, enzymes cannot reproduce independently of the living cells. In the intracellular phase, virion exists primarily as replicating nucleic acid that includes host metabolism to synchronize virion's components. Eventually, that is complete virus particle or virions are released. In summarized form, we can say the virus that differ from the living cells in at least three ways. Number one, their simple acellular organization. Number second, the presence of either DNA or RNA, but not both, in almost all virions of the uh, human being, except one virus that is called the human cytomegaloviruses, that is in a DNA genome, and 4 mRNA segments are also there. And third thing is that their inability to reproduce independently of the cells and carry out the cell division as prokaryotes and eukaryotes too. Okay, so first we will conclude about the total characteristics, what they are in the points. Just we can revise it, that virus, they are the metabolically inert particles, they are obligate intracellular parasites, virus cannot make energy or the protein independent of the host cells, virus multiplied inside the living cells using the host cell machinery. And along with all this information, that should I also want to discuss that viruses are also very site specific. That means only if their site is there, they will only capable to produce the disease, otherwise they will behave as an inert particle. Now in points, in short, virus as a living things, that virus occurs in the different strain. Virus have their own genetic materials. Virus can undergo the mutation. They get destroyed by the ultraviolet rays also. Now virus as a non-living thing, that they lack a cellular structure, they do not respire, they do not move and grow, they can be crystallized and stored in the bottles like a chemical, they lack enzyme system, do not have metabolic activity of their own. Now where the virus they lives, virus generally they are not able to survive other than host. So virus only can be in the active form within a host. Okay, and they are generally present in the animal cell, plant cells, and some bacterial cell also. Okay, they present either in the bacterial cell, animal cell, or in the plant cell. Examples could be the herpes virus, TMV virus, and bacteriophages. Because they are unable to reproduce independent in the living cells, virus cannot be cultured in the same way as prokaryotes or the eukaryote microorganism form. So they require some specific host within that they can survive. So different animal virus can um, inoculate in the embryonated egg also, that is fertilized chicken embryo, and that can be incubated up to the 6 and 4 weeks. So the different sites are the chorionolentic membrane, amniotic fluid, yolk sac, amniotic cavity. Within that, we can we can isolate the different type of viruses for the study purpose. Now, viruses are very less in the size than bacteria. Some viruses size are very long, can be up to the 20 to 1400. Maximum virus they belongs is the Poxviridae family, and the smallest virus that can be in the polioviruses is of 30 nanometers, that is poliovirus, and giant mimivirus that are also up to the 800 nanometer. You can see that different size of, you see that different size and shape of viruses. Now, different shape of the viruses. Virus can be having a different shape and size. They can be of filamentous, they can be of icosahedral, they can be of tadpole shape, and different type of genome could be there in the viruses. Now, what is a virus structure? Virus consists of the nucleic acid and the protein molecule, genome or the nucleic acid, and that is covered by the protein coat, that is called the capsid. Some viruses have an envelope that is outside the capsid. Generally, the envelope is coated with a host cell. On the envelope, some antigens molecules can be present, that is of the viral cell origin, and capsid without the envelope is called the naked viruses. You can see the structure of virus. See that envelope, capsid, and viral core. You see that icosidal symmetry within that capsid, that this. So all these single unit of the virus is called as virion. So all virions, even if they possesses other constitute, they are uh, that is of made up of by the nucleocapsid core. Some virus can only the nucleocapsid. Nucleocapsid is composed of nucleic acid, either DNA or RNA, that is held within a protein coat that is called the capsid, which protects the viral genetic materials and is aid in its transfer between the host cell. There are four general morphological type of capsid and virion structure. Some capsid are icosahedral in shape. An icosahedron is a regular polyhedron with 20 equal triangular faces and 12 vertices. These capsids appear spherical when viewed at the low power in the electron microscope. Second, other capsid are the helical and shaped like a hollow protein cylindrical structure, which may be either rigid or the flexible. Third one, many viruses have an envelope and outer membrane layer surrounded the nucleocapsid. Envelope virus have a roughly spherical but somewhat variable shape, even though the nucleic acid can be either icosahedral or the helical figures. Fourth type is called as complex viruses. They have a capsid symmetry that is neither purely icosahedral nor helical. They may possess the tail or other structures like in bacteriophages, having a complex multi-layered wall surrounded by the nucleic acid. Example of that is a pox virus, such as vaccinia viruses. Now, both the helical and icosahedral capsids are large macromolecule, that is a structure that constructed from many copies of one or few type of protein subunit, that is called the protomers. Probably the most important advantage of this design strategy is that the information is stored in the viral genetic material is used for the maximum efficiency. Okay. So about the microviral genome, it consists of either DNA or RNA. It may be single and standard form or double stranded. They are either circular or the liner. Now about the viral capsid. Viral capsids and viral nucleic acid surrounded by the protein coat, that is called the capsid. Viral capsid is of three types. First one is called the helical, icosahedral and complex symmetry. Okay. So first we will discuss about the helical. Okay. Helical symmetry. In this, the helical capsids are shaped much like the hollow tube with protein wall. The tobacco mosaic virus provides a well studied example of the helical symmetry. A single type of protomers associated together in the helical or the spiral arrangement to produce a long rigid tubes that is of 15 to 18 nanometer in diameter by 300 nanometer is long. The RNA genetic material is wound in the spiral and pointed towards the inside of the capsid where it lies within a groove formed a protein subunit. Not all helical capsids are as rigid as TMV capsid. Like influenza viruses, RNAs are enclosed with thin, flexible helical capsid folded within an envelope. The size of the helical capsid is influenced by the both of the protomers, that is nucleic acid enclosed within a capsid. The dimer of the capsid is function of the size, shape and interaction of the protomers. The nucleic acids determine helical capsid length because the capsid does not seem to extend much beyond the end of the DNA and RNA. The most suitable example for this is influenza virus and rabies viruses. You can see that. Now, second type of symmetry is called as icosahedral capsid, icosahedral symmetry, that is made up of icosahedral capsid that is of 20 triangular sides. Okay, this is a one of the nature's favorite shape virus that is employs the icosahedral shape because it is most efficient way to enclose a space. A new gene, sometimes only one, can code for the protein that self-assemble in form of capsid. Okay, so the icosahedral symmetry, that is virus that is negatively stained with viewed in the transmission in the electron microscope. A complex icosahedral capsid structure is very much common in the pox virus group, and capsid are structured from the ring or knob shaped unit that is called the capsomers, each usually made of 5 or 6 protomers, that pentamers, hexamers, and possesses a 6. Pentamers are at vertex or icosahedron where the hexamers that forms its edges and triangular faces. Okay, so this is about the icosahedral symmetry. Mainly the example of this kind of symmetry is HIV viruses, hepatitis B viruses and adenoviruses. Now, before discussing the hepatitis B, just I want to say about the nucleic acid. Nucleic acid, they are of virus, are extremely flexible that repeats the nature of their genetic materials. They employs all possible nucleic acid types: single strand DNA, double strand DNA, single strand RNA, double strand RNA. All 4 types are found in the all animal viruses. Plant virus most often have a single strand RNA genome. Okay. Uh, Although the phase may have a single stranded double DNA or single stranded RNA. Okay, so this is a specific feature of the vector phases. Uh, Size of the viral genetic material also varies according to their species. The smallest genome uh, is almost around 1 is to power 1 multiplied by 10 to the power 6. Okay, so most of the DNA virus, they are of double strand DNA viruses as their genetic material. Liner double strand DNA that variously modified found in the various viruses other than circular single double strand DNA. The lambda phase has an liner double strand DNA with cohesive ends. The single strand complementary segments, that is 12 nucleoside or tides long, that enables it to cyclize when they base pair with each other. Okay, so a single stranded DNA that is found in the parvoviruses. Double-stranded DNA is formed in the herpes viruses, vaccinia viruses, and polymerase viruses. While the single-strand RNA viruses, they are found in the picorna, rhabdo, and some retroviruses. While double-stranded RNA is also present in the reoviruses and some other polyhedrosis viruses. So this is the total just a structure of the hepatitis B. In this, icosidal symmetry is there, and genome is there, core genome is there, and mainly the virus is enveloped, and on the envelope there is some specific genetic arrangement is there, that is called the antigen, and these HBS antigens, they are responsible for the disease production. They are of viral coded. Now, the structure of HIV. Same structure of HIV that have an icosidal symmetry. In this, they as a genome, they have an RNA viruses, and with the RNA viruses, there is a reverse transcriptase enzyme is there. Mainly in the this is although the envelope virus made up of lipid content, and mainly the antigen which are appears in the patient serum are P24 core antigen, P7 GAG, GP120 and GP141. And one reverse transcript enzyme is also present that is useful for the RNA to DNA process. So the RNA genome is changed into the DNA genome, and that is more similar to the host cell. Now, this is a complex symmetry. The head of the virus is cubic and tail is helical. Examples, they are the pox viruses and bacteriophages. Now, the replication. The virus replication includes the two types. First is the lytic cycle and second is the lysogenic cycle. Lytic cycle includes the 5-step attachment, penetration, replication or nucleic acid maturation and release of the new virion. Now, the virus that takes place, the two type of cycle, that is called the lytic cycle and lysogenic cycle. Before discussing that, virus has divided. We have to just have an idea about that. That virus has divided to the different taxonomic group on the basis of the characteristics. They are related to the type of host, virus structure and composition, mode of reproduction, and the nature of any disease that they cause. Okay, some of most important characteristics are mainly they are nature of the host, that could be the animal, plant, bacterial cell, insect and fungal, the nucleic acid character RNA and DNA, and capsid symmetry icoside and helical and binal, and presence and absence of envelope, diameter of the virions and nucleic acid, number of the capsomere in the icosidal viruses, immunologic properties, whether they are immunogenic or not, genetic number and genetic map also, intracellular location of the viral replication, where the virus is enter and where its replication takes place, the presence or the absence of the DNA intermediate and presence of reverse transcriptase, the type of virus release and the disease cause and spiral clinical features, method to transmission. So, the life cycle, lytic cycle, in which the host cell genome is there, on which the virus is enter into the body and multiplies within the cell, and finally they will release their genetic material from for outside of the cell. So they uses the two type of cycle, that is first is lysogeny cycle and second one is of lytic cycle. Okay, so lysogeny cycle is a cycle that is of mild in the nature, in which the host genome is generally impregnated in the viral genome is impregnated into the host genome, and the replication takes place, and assembly and is very mild, and final release of the virus is also in the very mild way. Second is called as uh, lytic cycle, in which the reproduction of the virus is takes place in the very aggressive way, and the the viruses come out from the uh, cell by the very progressive manner and very destructive manner. Okay, you can see that diagrammatic representation of that. Now, the virus reproduction in the animal, that is of that is of different cycle, in which first is adsorption of the virion. That means first step in the animal virus cycle, that is adsorption to the host cell surface. This occurs through the random collision of the virion within a plasma membrane receptor site. The receptor is most often is a protein and frequently glycoprotein. So the several glycoproteins receptors have a sugar derivative salicylic acid attached, which virus can recognize. Sometimes a complex carbohydrate such as heparin sulfate is a receptor. Okay, according to the receptor, virus is attracted to that area, and then finally virus takes place, settle down to that type of area. Now, penetration of the viruses, that is called the uncoating. First, the whole virus can be engulfed, and sometime only the genetic material is penetrated. Okay, so virus penetrate the plasma membrane and enter to the host cell. Shortly after that adsorption, and the virus uncoating and removal of the capsid and release of the viral nucleic acid occurs during a short after penetration. Both events with reviewed together. The mechanism of penetration and uncoating must vary with the type of virus. That is because the virus differs so greatly in the structure and mode of reproduction. After encoding, the virus replicate, and final transcription process takes place in the DNA viruses. In early part of the synthetic phase, that is governed by the early genes, that is devoted to the taking over the host cell to synthesize the viral DNA or RNA. Some virus animal inhibit the host cell DNA, RNA and protein structure. Okay, so after the reproduction and transcription, finally this virus, they are released in the cell by the different way. Like they can release by the mild way, that is called the lysogeny cycle, and sometime they will release by the very aggressive way, that is called the lytic cycle. That by the lytic cycle they will release and come out to the next cell. Before that releasing, the different part of the, if they are different part of the virus, they are manufactured in the different areas, so they will assemble, and finally viral release by the mechanism of the virion. The differ between the naked and the envelope viruses. Naked virus appears to be released most often by the host cell directly, and by the lysis, and in contrast the formation of the envelope virus, the release of the envelope virus, there is usually um, uh, a process in which the host cells may continue the virion release for some time. So the first virus encoded the protein and incorporated into the plasma membrane, then nucleocapsid is surrounded by the release and envelope form of the membrane budding. So generally envelope virus, they will come out from the cell by the lysogenic process, and by the lytic process the naked virus they comes out. Okay, so conclusion is that virus are infectious particle with replicate only inside the cell. They attach plants, animal and bacterial cell. They replicate, exhibit a lytic and lysogenic cycle. They exhibit both the living and non-living characters. They have a called as origin at the age of the life. Now, uh, mm, at this point we had discussed about what are the virion, how they replicate, and what is their morphology, what is their classification, how they replicate, how they release. Now, we will discuss about the AIDS and the HIV viruses. What is the AIDS? AIDS is a acquired immunodeficiency syndrome that is of fatal illness, and that is caused by the retrovirus, that is HIV. Okay. The breakdown of the body's immune system that leave patients valuable to the host of the life threatening. Okay, and generally the infected patients, the HIV patient that is infected and that will change into the AIDS patient, generally the immune cell mediated immunity is good in their nature, but the mainly the immunity that is involved for the that is demonized, that is of cell mediated immunity, in which the CD4 receptors that are responsible for the this they deplete the T4 T3 cell, T8 T4 cell ratio is disrupted, or we can also say newly the new uh, nomenclature for the T cells are, what is it, that is of CD4 and CD8 cells. Okay, so after the infection of that, the ratio of CD4 and CD8 ratio is disturbed. So the patient, if they get infected, so they will not cure easily if they are HIV positive, or if they are attached to the what, they will, if they are attached to the infectious cell. So they are also called as retroviruses, the RNA genetic materials with reverse transcriptase, that is RNA directed DNA polymerase enzyme is remains within the viruses, and that is of spherical and envelope virus. You can see, check it. It contains the two identical copies of single strand positive sense RNA genome that is associated with the viral RNA is reverse transcriptase enzyme. The virus core is surrounded by the nucleic acid composed of protein. The virus contains the lipoprotein envelope. The major virus coded envelope glycoproteins are projecting spikes on the surface and the anchoring transmembrane pedicles, and these spikes bind to the CD4 receptors on the susceptible host cell. And viral genes, the HIV genome contains the three structural genes, that is GAG, POL and NO, that is characteristics of all retroviruses, five nonsense genes, and that is present on both HIV-1 and HIV-2, and HIV-1 contains the VPU and HIV-2 has a VPX. Now, epidemiology, male versus female, occurs in the all age group and ethics of the group. All areas of the country they are affected. AIDS is now the second leading cause of the death for all men's age, that is of 25 to 44 year age, and you can see that in India, Hyderabad is a most zone, and somewhere is in Goa, Bangalore, Mumbai, Nagpur and Chennai. Now, HIV agents, it's a RNA virus which replicate actively dividing T lymphocytes. Virus, they divides within a T lymphocytes, and that is of unique ability to destroy the T4. The T4 cell, what they are? They are helper cell. And reservoir, once the person get infected, virus remain in his body lifelong. And the person is symptomless carrier for the years before the symptoms actually appears. So the source is virus found in the great concentration in the blood, CSF and the serum, and low concentration have been found in the tears, saliva, breast milk, urine, cervical and vaginal secretions. They are generally also associated with the brain tissue, lymph node, and they are also found in the blood and the semen also. And they also appears in the cell tropism form, and like HIV infect the cell that expressing their surface of CD4, which is receptor for the viruses. Okay, and resistance, they are HIVs inactive by the treatment, with 35% of isopropyl and 70% of ethanol. You know that high risk people group are the male homosexuals, heterosexual partner, and drug abuse, blood transfusion and recipient, and hemophilias, and patients those having the STDs. As how they will transmit? By the sexual contact, by parental transmission and perinatal transmission, breaks in the skin, mucous membrane, direct injection. Route by the sexual contact, blood exposure, and perinatal, that is male to male. And these are the main route, injections and some other way. Mainly generally the operational, that is a healthcare worker, hospital staff, laboratory worker, and other rounds, organ transplantation, artificial insemination, and needle prick, that is also. Incubation period, that is HIV infected till the development of AIDS, few months to 10 years or even more. And uh, generally 75% of the person, those who are having the HIV, that will develop the AIDS almost within the 10 years. You can see that HIV virus, T cell, HIV infected T cells, and new HIV virus. You can see that new progeny is there that also cause the infection to the other cells. The clinical manifestation: initial infection, asymptomatic, AIDS related, and AIDS. And you can see actual acute HIV syndrome, 3 to 6 weeks following the primary infection, fever, weight loss, sore throat, malaise, headache, lymphadenopathy, rashes, nausea, spleen. You can see asymptomatic areas. And common manifestations of, if they change into the AIDS, common manifestations are lung infection, gastrointestinal infection, skin infections, central nervous system. Okay, so we can when find out the case of AIDS, we will confirm by the laboratory diagnosis. So then in the laboratory diagnosis, we are using ELISA test, rapid test, simple test, and Western blot tag for the screening. And we can also use for the different serological tests for their this screening test and confirmation. We will do the different type of confirmatory test like serology test. And prevention, the use of condom and contaminated syringe or needle should not be shared. Blood and blood products are screened before they are transferring to the others. So this is about the today's lecture. I hope you got information about the subject. So thank you very much for being with me. Have a good day.